Okay, so we left off here on Wednesday talking about how do we know which model of DNA replication is right. And there's what's called the Meselson and Stahl experiment. And what Meselson and Stahl did was they basically used new nucleotides that had a heavy form of nitrogen. And so what I want you to do is to work through this experiment, these two slides on your own, and to see how they demonstrated that the proper model is what Watson and Crick proposed, and that's that it's semi-conservative. That each strand in our old DNA molecule acts as a template for the new strand. Okay, because the rest of what we're going to talk about is all using this semi-conservative model. That's the correct one, but this experiment shows you how they determined that it was the correct form of DNA replication. All right, our next question. How does the new strand of DNA get written? How do we actually write that new strand, okay? Because it's one thing to say, all right, well, each strand from our original molecule acts as a template to write a new one. And it's one thing to say that it's complementary, right? So you can only fit one base in a particular position. It's a whole different question to say, how does it actually happen? Because I'll tell you this, it is not spontaneous. Your DNA does not spontaneously replicate itself. It is a highly controlled, highly efficient process. Okay? And so here's what basically some of the steps involved. First, we need to unwind the DNA or separate the two strands from one another. Okay, we need to unwind the DNA, separate the two strands from one another. And in order to do that, what types of bonds do you need to break? Hydrogen bonds. Good. And breaking hydrogen bonds is fairly easy. They individually are weak bonds, okay? And there are millions of them, but if you're only breaking them on one side, it's like unzipping a zipper, right? To rip a zipper apart is challenging. It's not impossible, and it's actually pretty cool to do it, but it's challenging, right? But to unzip it, break those links one at a time is very simple. Very simple. It must be because my children can do it. Oh man, the backhanded compliments. Oh, I love it. All right. DNA polymerase adds nucleotides to an existing chain. Okay, so a very simple answer to this question of how does the new strand of DNA get written would be for you to say DNA polymerase adds new nucleotides. Okay, adds new nucleotides. So then you're like, cool, DNA polymerase is a machine that is reading the old strand, reading one of those templates, and writing the new strand. And you're like, that's so cool. And so when you look at DNA polymerase, it's a hand-shaped protein. It looks like a hand like this. You're like, hands don't normally look like this. You're right, but they do when you're catching a Frisbee or eating a sandwich. Two of the best things you can do as a human being, right? I mean, just eating a sandwich is obviously the better of those two. But DNA polymerase looks like this, and when you look at the protein, you find that it's a protein with quaternary structure. And what does that mean to you? And it's the beauty of it, because what it needs to mean to you is the truth, right? So what does that mean?
It has two types of
At least two types of proteins interacting together to serve a single purpose, and that is writing the new strand. Okay? So we have multiple proteins that are all combined together to build this DNA polymerase. And I forget how many there are, but it's more than two. I want to say it's like 15, but for some reason that number's in my head. You don't need to write it down because it might be wrong. But so these various domains are built to do certain tasks because DNA polymerase needs to add nucleotides, and we call these deoxyribonucleotides because what's the sugar in DNA? Deoxyribose. Okay, so we because ATP is so common, when you say nucleotide, it's usually assumed that you mean ribonucleotide. Okay, because there's so much ATP, that's a ribonucleotide. So we specify that we are adding deoxyribonucleotides, but only at the 3' end. When you see this, you need to note something that is very significant about DNA polymerase. Really two things. One, it can only write in one direction. Because if you can only attach a new nucleotide at the 3' end, it means you cannot attach it at the 5' end. Yes? Which means you can only write in one direction. That's a very significant thing to take away from this sentence. Who knew that you can take multiple things away from a single sentence? It's a sign of a well-written sentence. You're welcome. Ah, see, there it is. On Friday, the smiles come a lot more freely because you're like, 45 minutes from now, I'm done, right? I'm going to go get myself a sandwich and a Frisbee and not think about DNA polymerase, even though it's the same shape, right? So we can only add them at the 3' end. That's one thing that you need to see from this sentence. Another thing that you need to see from this sentence is DNA polymerase cannot attach the first nucleotide. Notice it's attaching deoxyribonucleotides at the 3' end. If there's no nucleotide to start the process, there is no 3' end yet. Right? So that means DNA polymerase cannot start this process. Or it's what we say is it is not self-priming. You actually need a primer. And this used to be a great analogy when you needed like a primer to start an engine. But engines now are all like a mix. Yeah, they're highly controlled and it does all the mixing for you, right? It mixes the oil with the gas and it's just all, it takes care of all of that for you. But so you need a primer, you need something to start the process, and then DNA polymerase can take over and start putting in new DNA nucleotides at the 3' end. David?
Can you repeat the second one again? Sorry.
Yeah, DNA polymerase cannot add the first nucleotide. It can't add the first nucleotide. It can only write in one direction, and it cannot add the first nucleotide. Those are both very significant truths to take away from this one sentence. Okay, so we have a domain for adding new deoxyribonucleotides. There's also a domain for reading the template strand. And it can only write by adding new nucleotides at the 3' end, keeping in mind that the strand it's reading is anti-parallel to the one it's writing, right? We know DNA is anti-parallel, so if it's writing 5' to 3', attaching new nucleotides at the 3' end, it has to read backwards of that. It has to read 3' to 5', because again, the two strands are anti-parallel. This one runs 5' to 3', this one runs 5' to 3' this way. Okay? So if it's writing this way, it's reading this backwards. Okay? So it has a domain for reading backwards and then writing forward. And you're like, that's so cool. It'd be like if you were to go and copy something that a classmate gave you and you're copying it, but in order to do so, you have to read it backwards while writing it forward. You're like, I just copy and paste. I don't even worry about retyping it. Like, it's so simple. It's not that simple when you're polymerizing DNA. And then the nucleotides we know are added according to Shargov's rules. Okay, if there's a T on the template strand, it has to add what? An A. It has to add an A on the new strand, because anything else will not work. It will not form hydrogen bonds, and so it'll create basically like a speed bump for this machine trying to truck its way down the DNA molecule. This is a massive machine. Massive machine. Y'all seen the original Cars movie? This is her name Bessie, the asphalt machine. I mean, this is Bessie. I mean, this is a massive machine. Okay. So here's this process. All right. So here is our old strand. Okay. Here's our old strand, our template strand. Here is the new strand being written by whom? Or by what? Because it's not a person. By DNA polymerase. And so it's going to attach new nucleotides at this 3' end where we have a free carbon 3. Okay? And so it will come in here and it will read the complementary strand and it sees a T there and it will bring in a adenine deoxyribonucleotide. Okay? And when it does, look at it. It came in with 3 phosphate groups, but by the time it adds it only has 1. And so you're like, that's so cool because I know when I break phosphate groups, what happens? It releases energy. And then that energy is used to build new bonds. And you're like, that's so cool. The exact materials we're using to build this are also providing the energy necessary to carry that out. Be like, you have this factory and you make, the goal of your factory is to make bags of hot Cheetos, right? No other chips, because no other chip compares to the deliciousness of a hot Cheeto, right? But it's like the hot Cheetos, as they come in, are also providing the energy necessary to run all the machines. So like even the machines love to eat hot Cheetos. That is just a beautiful, beautiful thing happening. Okay, so here's the hand of DNA polymerase. And again, we've got domains for reading the template strand. We've got domains for attaching the new nucleotides. And it really does look like a hand. And so now every time you eat a sandwich, you'll probably think about DNA polymerase. But then knowing that knowledge has a half-life, it'll really be just for like the next 3 hours. If you eat a sandwich, you'll think about DNA polymerase. After that, it'll be like, DNA who? Levi.
Okay, so the
DNA what? Because it's not a person. I keep
fingers are
persona.
labeled as fingers.
Oh, yeah.
Well, what are they?
There are other protein domains.
Okay.
So remember, this is a protein with quaternary structure. So there's like one protein here making up this finger, one protein here making up this finger, one protein here. And it's like, because we tend to oversimplify this process. And we're like, all it's doing is it's bringing new nucleotides in and it's just putting them in place. But no, it has to hydrolyze these bonds, right? It has to cut these bonds with water. So you have to have one of these fingers that can hydrolyze these deoxyribonucleotides, hydrolyze the phosphate groups off. You have to have another finger that can actually create the phosphodiester bond. You have to have another finger that can read the template strand and ensure that the proper one is written. You have to have another domain that can check for errors, and if there are errors, to back up. And then you have to have another domain that when you back up, you cut out the nucleotide that you added incorrectly. And it's like all of these different jobs are accomplished by a different protein making up this massive machine. And you're like, Dr. Engel, this isn't exactly like Bessie, because all she did was basically heat the asphalt and then drop the asphalt, right? Because this is a much more complicated machine. You're absolutely right. This is like a 3D printer printing houses. Have you ever watched that? A 3D printer print a house? It's pretty cool. There are places where they are 3D printing houses out of concrete. And it's pretty gnarly using 3D printers. Or you know what's really cool is to watch a 3D printer print another 3D printer. Like it's reproducing. And then if you watch a 3D printer print a smaller 3D printer, you're like, oh my gosh, it's adorable. Who knew? Who knew that 3D printers and sandwiches would be part of our Friday class? I didn't know. These things just come to me. All right. So what other proteins are involved in DNA replication? So far, we've only talked about one protein complex, and that's DNA polymerase. And you know it's made up of multiple protein subunits, but we really only talked about one protein with quaternary structure. So let's talk about some others. We've got a protein called the sliding clamp, and it looks like a donut. It wraps around the template strand and attaches to DNA polymerase to keep DNA polymerase attached to the template strand. And you're like, oh, this is like whatever attached Bessie to Lightning McQueen. Kind of. But this, in order for the analogy to really work, Bessie would need to be attached to the road, and Lightning McQueen would need to be the sliding clamp that kept her attached to the road. You're like, your analogy is breaking down. Stop trying to sell it. Okay, so that's another protein, the sliding clamp. We have a protein called DNA Helicase, and it breaks the hydrogen bonds. It comes in and unwinds the DNA strands by breaking hydrogen bonds. That's called DNA Helicase. Yeah, Micah.
Helicase comes before polymerase, right?
They're all attached together. So helicase is the first protein to show up, typically. That's actually not true, because there's a molecule that's called DNA-A, and it's the first one to show up. But this is more detailed than what you need to know. It's the first one to show up, and it breaks the first set of bonds, and then helicase can come in and start unzipping it. So helicase is like the first charismatic person that shows up, right? You know, because we liked, I'll use the analogy of an athlete. We're like a good athlete, like a quarterback, right? If he's going to be a good quarterback, he's got to be the first one in and the last one to leave, right? Just got to be a leader. But the reality is, no, there are already people there. They're just like the silent, the silent partners. They're not the leaders, but there are already people there. Think the quarterback's beating like the janitors in? No. You're like, what about if it's like Johnny Manziel? He didn't even show up. You're right. Now he's no longer a quarterback in the NFL. Yeah. Sorry. Not to Johnny Manziel, but to you for using him as an illustration. So Helicase. Now what ends up happening is Helicase attaches to DNA polymerase, and this whole machine is just all attached together. We have single-stranded binding proteins, and these keep the two strands from reattaching, the two old strands from reattaching before you have a chance to write the new strand. Because Helicase is coming in, it's breaking the two strands apart. These single-stranded binding proteins hold on to those two lonely strands to keep them from coming back together, because they need to be a template, right, for a new strand. Topoisomerase, also known as DNA gyrase, two names for the same protein, topoisomerase or DNA gyrase. When you start pulling these two strands apart, you can create supercoils. If you ever tried to unbraid hair by going at the top of it and just pulling them apart, totally doesn't work. And it creates a supercoil underneath and above, which I'm sure is not pleasurable. I don't know. I've not ever had hair long enough to braid it. But it's like when you go to unbraid somebody's hair and you try to pull it apart in the middle, it creates supercoils. So what topoisomerase or DNA gyrase does is comes in and cuts the strands downstream of that to relieve those supercoils. And then here's the one that starts the process, primase. Because we know you took from that sentence, DNA polymerase can only write in one direction, and DNA polymerase cannot add the first nucleotide. Well, primase does. But look at this. Primase writes a short RNA primer, so it uses ribonucleotides, and then DNA polymerase comes in and starts doing deoxyribonucleotides. You're like, why would it do that? Why wouldn't we have something that writes DNA nucleotides? We do in our cells, in eukaryotes, but prokaryotes actually write an RNA primer. Actually, that's not true, because we have a DNA polymerase, but it still writes an RNA primer. I just told you something that was wrong. I'm sorry. But even in our cells, we start with a short RNA primer, and then DNA polymerase takes over and puts deoxyribonucleotides. You're like, that's so weird. Why would you do that? Because it can't stay like that, right? Because otherwise you're going to have these hybrid DNA, RNA molecules in your nucleus instead of DNA, and every time you replicated, you'd get more and more RNA showing up. But that's not what happens. So eventually these have to get cut out, these RNA primers, and replaced with DNA primers. You're like, this is so weird. Why would they do that? It's a wonderful question. One that I don't know the answer to. I have some speculation, but I don't know the answer to. Chris.
Why can't the DNA polymerase start it on?
Yeah, that's an excellent question. It does not have a protein domain that can add one if there's not a 3' carbon there to add it to. It just is physically incapable of doing that.
So once the 3' exposed? Then
once you have an exposed 3' in, then it can add new nucleotides. Yep. Now primase can do that. Primase can add the first one. It doesn't need a nucleotide to already be in place, but it requires a different protein that can do that. Yeah. Okay. Couple more proteins. Ligase attaches fragments together. As you are replicating DNA, you end up getting to the end and getting a point where you have a free carbon 5 sitting right next to a free carbon 3, but there's not a phosphodiester bond connecting them. And we'll see this in a couple of slides. I'll show you an image and you'll see why this is the case. Ligase comes in and makes that phosphodiester bond. Makes that phosphodiester bond. You'll see. You'll see why that happens. And then one more protein, telomerase. Telomerase prevents chromosomes from shortening at the end. Okay, because when you replicate DNA, every time you replicate a chromosome, it shortens a little bit at the end. And again, you'll see why that is in a minute. Telomerase is a protein that will come in and fix that shortened chromosome. It'll lengthen it back. So, have I talked before about 23andMe and Ancestry.com? You can send in a saliva sample and they'll tell you your ancestry as best they can, by matching it to genetic structuring across the globe. There's another company, and I forget what it's called, but you can send a saliva sample in and they'll tell you how long your telomeres still are. Because a lot of what determines the aging process is the length of your telomeres, the ends of the chromosomes. Because every time the chromosome replicates, it shortens a little bit. And eventually you shorten past the telomeres where there aren't any coding genes, and you start losing parts of your chromosome that code for things. Okay? And it basically, you get to a point where you go into senescence, that the cell can no longer function properly. And so you can send your saliva in, and it'll tell you how long your telomeres are, and then compare that to the average. So like if you're 19 years old and you send it in, they tell you that you have the telomeres of a 35-year-old, then you're like, well, I guess I'm probably not living to 77, right? Or whatever the average human length is. It's like a death clock. Have I talked to you before about the death clock?
Last semester.
Okay, last semester. I'll mention that again because it was just such a cool thing when I was a teenager. I mean, it told you exactly when you were going to die. I mean, how cool is that? And then counted down from that moment. All right. So here is the sliding clamp holding our DNA polymerase, our hand-shaped machine onto the template strand so it can continue to read it and continue to add new nucleotides on the coding strand. Okay, here is Helicase. Okay, Helicase is right here. Okay, coming in, breaking hydrogen bonds, separating the two strands. Here you have single-stranded binding proteins, keeping those two strands from recombining before DNA polymerase can come and write the new strand. And then here we have topoisomerase, also known as what? DNA gyrase. That's over down here, eliminating supercoiling. Oh, and then here we have primase, starting the process by writing a short RNA primer that then DNA polymerase can take over. And look, we still have those RNA nucleotides in there. We show them in blue and the DNA nucleotides in red. And I already told you, this can't remain. Because otherwise, every time you replicated the DNA, there'd be more and more RNA showing up, and eventually it'd all be replaced with RNA. But that's not what happens. So something has to come in and cut this out and replace it with DNA nucleotides. Micah, then David, then Chris.
What cuts it out?
Another RNA polymerase, specifically RNA polymerase 1. So in bacteria, there are at least 5 different DNA polymerases. DNA polymerase 1 through 5. Okay? In eukaryotes, there are at least 16 DNA polymerases. And we don't call them DNA polymerase 1 through 16. We get fancy with it, and we do DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, epsilon, so on until you get through 16 letters of the Greek alphabet. I don't know that many. I don't know them in order, so I can't share them. David.
So does the RNA primer, does it create a 3 prime? So then
You mean, is it exactly 3 nucleotides?
Yes.
No, usually it's more like, I think it's more like 10 to 25 nucleotides of RNA.
So without the RNA primer, the new DNA.
Yeah, DNA polymerase can't start the process. So without the RNA primer, DNA polymerase can't attach nucleotides. But then it's like, well, why isn't there something that can just write a short DNA primer and then take over? That's an excellent question. I can tell you some advantages to this, but I don't know why it's this way other than to say that it seems to be the way God made it, because all organisms replicate their DNA like this. They write a short RNA primer, DNA polymerase takes over, another DNA polymerase comes in, cuts out that RNA primer, and replaces it with DNA nucleotides. So it's conserved among all living organisms. Yeah?
Why can't we use DNA as a primer? Can you answer that?
Well, there's nothing that writes DNA as a primer, is the thing. There's no machine that writes a DNA primer. There are machines that write an RNA primer, but no machines that write a DNA primer. So why can't we? There's just no machine to do it.
Okay. It's interesting because it seems like it's not efficient with energy.
Yeah, except for, then to consider that replicating the DNA is not the only thing a cell does, right? The cell also writes DNA into RNA, so it's using a machine that works doing something else. So it actually creates some nice efficiency.
Okay.
Yeah. And then so here's the whole thing together, and even this is a little bit misleading because these are all physically attached together. Helicase is physically attached to the DNA polymerase, and there's actually two DNA polymerases, one on this strand and one on this strand. One that's writing this strand and one that's writing this strand. There are two DNA polymerases, both attached to helicase, all of them speeding down the chromosome, writing 1,000 bases a second. Yeah, that's a lot. 1,000 bases a second, and it's all speeding down. I mean, this was like when Lightning McQueen tried to do the road real quick and take like 5 minutes, right, and just drove it down, built a terrible road, right? You know what I'm talking about? And then he had to go and do it slowly. But this works super fast and makes very few errors. Kind of cool. I mean, it's really cool. It's not kind of cool. It's really cool. So here is telomerase. Oh, I didn't get to you. I'm so sorry, Chris.
No, mine was the same as David's.
Okay. But did you have another question?
I'm still there.
Okay. So telomerase, we got to the end here, and basically this ended up, this whole section here was lost, and we'll see why that is in a minute. And it really has to do with, because you can only write in the 5' to 3' direction, right? So we have an RNA primer that sits here, primase writes it, but then when DNA polymerase comes and cuts it out, it has no free carbon 3 to add a nucleotide to, right? There's no free carbon 3 here, and so you just end up losing this information. Unless telomerase can come in, can recognize here, write this out a little bit longer, and then that way you have some more room to get a RNA primer in there and get DNA polymerase to get a lot more of that. And so here's that process. You can see it's here, we wrote a new stretch of DNA. Now we had some more room to write the RNA primer further back and get DNA nucleotides to make up more of what was lost. Now, we do not have telomerase active in all of our cells. Okay, most of the cells in our body, telomerase is not active because it's not good to have immortal cell lines. When you have immortal cell lines, they accumulate mutations and then they just keep passing those on as they accumulate them. So you only have telomerase active in areas where you need to basically restart the clock every time you have a cell division. And where might that be in the human body? We'll take a lecture break after this, but I just want you to think about this real quick. Where in the human body and what tissues do you want to restart the clock whenever you make a new cell? Skin cells is one. Where you have actively dividing skin cells at the base of your skin, you basically want to restart the clock as they move their way up. And you're like, why? They don't live very long. But it's you do. Anyways. And the other one is when you're making gametes. Because otherwise, if you made gametes and you didn't lengthen the chromosomes, your offspring would have shorter chromosomes than what you started your life with, right? That's not good, because that means every generation would live shorter than the one before it. Okay, but telomerase is very active in the tissues where you're making gametes to keep basically restart the clock. All right, so lecture break time. What I want you to do is this. I want you to draw the two old strands forming the double helix, and at one end of it, they are separated, okay? Right? So can you picture that in your head? You've got two strands at one end, they're still connected together, the two old strands. At the other end, they're separated, and you are writing the new strands. And I want you to illustrate where we're going to have a problem because of the only direction DNA polymerase can write. Okay? So in order to do that, what you're going to need to do is this. You're going to need to have your two strands. Wow, that's really light. Can you even see that in the back? I'll press a little bit darker. And so we've got our 5' end and our 3' end, and then here is 5' to 3', and we are writing new strands. Point out where you're going to have an issue because of how DNA polymerase writes. Okay? And then we'll pick up here in 60 seconds, 90 seconds. How long? 90? 90 seconds. Did somebody say 80? 90 seconds starting now.
Like I would wake up and like, oh, that me? Huh? No. Oh, okay. My body. No, it's not even like. I don't say it's just like the things in my body.
Just draw that.
It's all here. I'm just doing it's all my body. Yeah.
Well, they shouldn't because there's a Helicase right here.
I think it's
True, but there's still an inflammation.
it seems like
They're going to. Oh.
Got about 10 seconds. All right, anyone figure out an issue? So DNA polymerase can only write in one direction, and that's in the 5' to 3', and it can only read in one direction. And remember, helicase is here, and it's unwinding the DNA here, right? And then you have a DNA polymerase attached to it for this strand, and you have a DNA polymerase attached to it for this strand. Okay? This one runs 5' to 3'. So if the machinery's moving in this direction, what direction would you need to write it? 3' to 5', right? You'd be having to read 5' to 3', and you'd have to write 3' to 5'. DNA polymerase can't write in that direction, but the whole complex is moving that way. Like, what's happening? How does that even work? Now this one, no big deal, right? Helicase again is moving this way. There's a DNA polymerase attached to this strand, and it's moving this way, and it's reading 3' to 5' and writing 5' to 3'. No issue, because that's the way DNA polymerase writes. But you have another DNA polymerase that's moving in this direction, but it can't write in that direction. And you're like, how on earth can that happen? How can it move in this direction, but write in this direction? Right? It's moving in this direction, but it has to write backwards. Does this seem weird to you? It should seem weird to you. You're like, this is like walking and chewing gum at the same time, right? It's just challenging. That's what they say. I've never actually paid attention to if I can walk and chew gum at the same time. I think I can. I think the issue is really trying to chew at a different rhythm at which you're walking. Because that, I think, would be more challenging, right? Because I think what you'd find is you'd be chewing in sequence with your steps. Okay, and so what we get is what we call continuous replication and discontinuous replication. So our next question is perfectly out of what you just did for your lecture break. Because this one, again, the machinery's moving this way, this DNA polymerase is moving this way, no problem. It's going to do continuous replication. This DNA polymerase is moving this way, but it can't write this way, so it's going to have to write discontinuously. And what you find is that it actually pulls the strand through backwards. This DNA polymerase that's moving in this direction on this strand, it pulls this through backwards. So even though it's moving forward, it's pulling the strand through backwards. It's as though it were moving this way, even though it's moving this way, because the strand is being pulled through backwards. I'll show you. You're like, what does that even look like? How does it even work? Let's get through this slide real quickly, and I'll show you some diagrams that will show this. So you need to remember that DNA polymerase can only write in one direction, 5' to 3'. And that is because it can only attach a nucleotide where there is a free carbon 3, right? Which is going to be at the 3' end. And if you're writing at the 3' end, you are writing 5' to 3', right? Right? Okay. You also need to remember that the two strands are anti-parallel, which is illustrated right there on the board. And you're like, well, if they were just parallel, we wouldn't have this problem. You're right, but then the bases wouldn't hydrogen bond and they wouldn't hold together. And you're like, well, that eliminates the good thing about a DNA molecule, but they're parallel. Yeah, but they don't stick together. They actually repel each other. It's a problem. And then you have to remember that helicase unwinds the two strands at a particular place. We call this the origin of replication. And this creates a specific problem, one that you just illustrated during your lecture break time. And that is one strand can be written continuously by specifically DNA polymerase 3. This is in prokaryotes, right? Because I told you eukaryotic DNA polymerases are labeled with what? A Greek letter, right? So like DNA polymerase alpha, DNA polymerase beta, gamma, delta, epsilon, and I don't even know what comes after epsilon. Anyone know? I don't know. The other strand has to be written discontinuously in fragments that are called Okazaki fragments, as the other strand is pulled through backwards. And these are written primarily by DNA polymerase 1. DNA polymerase 3 is still at work, but you have DNA polymerase 1 is at work a lot more on the discontinuous strand. Sometimes we call that the lagging strand. It's an unfortunate word because it makes you think that it's slower than the other one. It's not slower, it just writes it discontinuously. Okay? So we call it leading and lagging, which I think is unfortunate. It'd be better to call it the continuous and the discontinuous. Chris?
So I'm kind of confused. So for that first bullet, it says remember the DNA polymerase writes in the 5' to 3'.
Mm-hmm. It reads 3' to 5', but it writes 5' to 3'. Because it can only attach a nucleotide at the 3' end,
Okay.
right? And if you're attaching a new nucleotide at the 3' end, which side are you growing, the 5' or the 3'? Now, if you're writing at the 3' end, you're growing the 3' end, right? Which means that you're writing 5' to 3'. You're growing the 3' end. Yeah.
So on the picture, the bottom strand would be the one.
The
that's
bottom strand
continuous?
is the one that you can write continuously.
Okay.
The top strand is the one where you'd have an issue. Okay.
So it reads 3 to 5 and writes 5.
Yes.
Okay.
Yep, absolutely. Now, one thing you need to know that we haven't talked about yet is that DNA polymerization actually proceeds in both directions. So if you looked at this DNA molecule, this would continue all the way over to here and it'd come back to here, and you'd have DNA polymerase happening on both sides of this, forming what we call a replication bubble. And again, I'll show you an image of this. You'll see it. You'll be like, wow, it really does look like a bubble. And then you'll appreciate why they call it a replication bubble. You'll smile. You'll think about, using one of those big, massive wands and running through a field making massive bubbles. Do you still enjoy playing with bubbles? I loved it as a kid, and I thought I wouldn't like it as an adult, and then I had children. I'm like, man, I still love making bubbles. It is fun. It's just, I don't know, it's just so simple. And I even enjoy chasing the bubbles, right? And then they pop, and it's just, man. That picture is going to be in your mind along with DNA polymerase holding a sandwich, right? Dr. Engel running through the park chasing bubbles. I don't run very much, so I'll chase a couple of bubbles and then I, sit and watch the kids do it. That you can probably imagine. All right. So here's the continuous and the discontinuous replication. Okay? So again, we've got helicase sitting right here, and it is physically attached to this DNA polymerase on this strand, and it's physically attached to this DNA polymerase on this strand. However, this DNA polymerase is writing in the order it's moving, but this one has to write backwards from the direction it's moving. The only way it can do that is by pulling the strand through backwards. So it basically lets go. Say it'll write this fragment here, okay, backwards by pulling this through backwards. So it's moving in that direction. It pulls this little section through backwards, writes it. Then it lets go and reattaches way up here and pulls this section through backwards, okay? And then lets go and attaches further down and pulls that section through backwards again, writing short fragments called what? Okazaki fragments. Why? Because they were discovered by a man named Okazaki. I know. Yeah, like those are unfortunate when people name things after themselves, because then it doesn't really make sense. You know, it doesn't convey the meaning in its name. But I'm not gonna lie. If I discover a parasite, I'm gonna name it after myself. Why? Because everybody should have a parasite named after themselves, right? Either yourself or one of your children. I might actually name it after my son, you know, because I love him so much, you know. Wouldn't that be a horrible thing to do to your kid, to name a parasite after them? Oh, I would never do that. Not to my son, maybe to one of my daughters. Oh, I'm totally kidding. Gosh. I'm gonna bring my kids when they're on spring break, so you get to meet them. At least the older two. I won't bring Madison because she can't sit still quietly. Reagan can't really either, but she'll whisper. So, okay. So here, and again, they've drawn this for simplification, but it is not accurate because Helicase, again, is physically attached to this and physically attached to this. So this is showing you DNA polymerase actually moving backwards. It doesn't move backwards. It's moving that way. Okay? Right? It's moving that way. What it does is it pulls the strand through backwards. Okay? Because it's moving that way and it has to write backwards, so it pulls the strand through backwards and writes. That make sense? Yeah. Okay, but what you end up getting is when you pull this through backwards, you get this big old DNA loop forming on the other side of the DNA polymerase as it's being pulled through backwards, and it looks a little bit like a trombone. You know what a trombone is? It's got that slide, you know? And so it looks a little bit like a trombone, and they call it, what do you think they call it? They call it the trombone structure. I know. Okay, but another issue, so we're like, okay, we've got continuous replication. This one has to do discontinuous because it's got to pull the strand through backwards, then let it go and grab further upstream and pull it through backwards. We're like, okay, that's a little weird, but I think I can picture that. But then we've got another issue. We got DNA polymerase 3 is the main polymerase. You see that, DNA polymerase 3? It's the main polymerase, DNA polymerase 3, still going on this lagging strand, where it's writing here, but we need to get rid of this RNA primer. That's where DNA polymerase 1 comes in. Does it even show this? It doesn't even show it in the next picture. Why do I even have the next picture? This is terrible. It's just terrible. Okay, so anyways, we've got an issue. DNA polymerase will write all the way up until it gets to the primer. So when this is like pulling this strand through backwards, and then it lets it go and grabs it like further up here when this opens up more, right? And then it'll pull it through backwards, and it'll write all the way until there's that RNA primer. The DNA polymerase will write all the way up in this Okazaki fragment until it gets to where that RNA primer is, and then it'll let go. Because DNA polymerase 3 cannot cut out this primer. That's where DNA polymerase 1 comes in. DNA polymerase 1 can cut out the primer and replace it with DNA nucleotides. Okay? And it'll do that. It'll come in, it'll cut out this RNA primer, so it'll pick up where DNA polymerase 3 left off, right, up against the RNA primer. It'll come in there. As it's writing the DNA nucleotides, it's cutting out the RNA nucleotides. But when it gets to the end of that primer, it can't attach the two together. Because DNA polymerase 1 isn't capable of doing that, attaching the two together, and that's where DNA ligase will come in. DNA ligase will come in and will fix that nick. So you see in here, I guess it did kind of show you because it cut out this RNA primer and then it opened up more and primase made another one and the DNA polymerase 3 pulled this through backwards and wrote it, right? And then it shows you like this is cut out, and you know DNA polymerase 3 can't cut that out, so it must have been DNA polymerase 1 that came in and cut out that RNA primer. But then we're left with a nick at the end. After it's replaced all the RNA nucleotides, ligase will come in and will create that phosphodiester bond. Fix that nick. Okay? You're like, wow, there are a lot of machines involved in this process that is not very simple. I don't know if I like this. But it happens. And so you kind of just, you got to deal with it, even if you don't like it. It still is not showing us DNA polymerase 1. It's awful. This is so bad. And again, it's still showing us that the DNA polymerase is moving in this direction. That's just for simplicity. You know DNA polymerase is actually moving in that direction, and it's pulling the strand through backwards, right? You know that, even though it's illustrated otherwise. Chris?
Does the DNA polymerase for the leading strand or the continuous strand does it ever like outrun the
No, not really, because they're all attached together. They're both that's a good question. Both DNA polymerases are physically attached to Helicase. It's all just one big machine. So it can't outrun it. And that's why, like, we'll call this the lagging strand, but it's not. I mean, is it slowing the process down? Maybe, but everything slows down as a result, right? It's like the whole mantra, like, you're only as good as your weakest player, right? You heard that with team sports. You see it a lot in football. I don't think you see it as much in some other sports because you can get away with only having like 5 good players in basketball, right? They're going to get tired at the end of the game unless their conditioning's really good. But in football, you've got minimum 11 people, and that's if you're playing both directions the whole game. And so there you're really only as good as your weakest link, you know? Really only. I mean, it happens in basketball too. I don't want to say it doesn't happen in basketball. You know. Yeah. Okay. Any questions? So continuous, discontinuous. Here's the replication bubble, keeping in mind that basically there's a helicase with its 2 DNA polymerases moving this direction. There's a helicase with its 2 DNA polymerases moving in that direction, creating the replication bubble. Now notice you've got a leading strand or a continuous strand and a lagging strand on both sides. But notice which one is the leading strand and which one is the lagging strand is reversed based on which direction you're going. This one's proceeding to the right, this one's proceeding to the left, so it should be reversed, right? Because right and left are opposites, right? Okay, so if this one is moving to the left, the one that you would write continuously to the left is this strand. The one that you would write discontinuously to the left is this one. But over here, this one's moving to the right. The one that you would write continuously to the right is this one. The one that you would write discontinuously to the right is this one. Yeah, Micah.
In the middle, wouldn't there be 2 carbons that match up that can't bond together, like 2 5 primes?
Yes, so ligase will come in. Oh, you're talking about right here?
Yeah.
No, so right here, because you'll still have 3 prime, you'll get this one will write until it runs up to that primer. Right?
Oh.
DNA polymerase 3 will go all the way up until that primer, and that you'll have a free carbon 3 there and the carbon 5 on this side. And then DNA polymerase will come in and cut out that primer. DNA polymerase 1 will cut out that primer, replace it with DNA nucleotides, and then you'll get that nick there where you'll have a free carbon 3 and a free carbon 5, and then ligase will come in and create the phosphodiester bond. Yeah. I know. We'll take some time in lab on Monday and Thursday of next week, and we'll write this out, because those of you that were there on Thursday, you heard me say this. I didn't say this on Monday. Next week's lab takes a long time. It just does. You're like, you had lab this week, and you're like, oh sweet, it took like an hour, maybe an hour and 10 minutes, and you're like, this is the new norm, right? We're gonna be done. It's not the new norm. The lab next week, it takes a long time because we have to set up a gel and let it run for like an hour plus. And so it's like during that hour plus, I'll be like, hey, well, it won't work on Monday because our lab doesn't start till 1:55. But on Thursday, I'll be like, hey, go grab some lunch. On Monday, I'll be like, I'm sorry, you can step outside and, eat a cookie or something. But then what I'll do is I'll actually draw out some of these processes during lab next week and just help to make you more comfortable with them, right? Questions, thoughts, concerns? So we're in a position where we have to make one of 2 choices. We still have 3 minutes of class left, but we're in our next set of 4 questions. So I want to present something to you. Do you want to start in on the next 4 sets of questions, or do you want to end class 3 minutes early? If you want to end class 3 minutes early, show it by a show of hands. Motion carried. Goodbye.